Can you hear me? Okay, yeah, thanks. I think that's better now. Hello, everyone. It's an honor to be here and be able to share and discuss these wonderful projects here. Today, I'm going to tell you about some smaller study which we did in North Mexico, and it's focusing on the insect distribution along elevation gradients in Mexican transition zone. Biologists have been obsessed for over 100 years with studying species along elevation gradients. So do we really need another study along altitudinal gradient? Maybe not necessarily, but what makes this, oh, sorry, I just tried to, what makes this research or study interesting is that it's situated in the biogeographically interesting zone. We studied insects in the Sierra Madre Oriental, which is the chain of mountains connecting central and South Mexico with the north. And it's a part of this so-called Mexican transition zone, where biotas of different biogeographic origins meet and interact. And we've been doing this study with my wonderful colleagues and students. And the project site is located in the state of Tamaulipas, which is here in that red dot. So the Mexican transition zone is basically a region where we can see the mixture of environmental or elements from both Nearctic and Neotropical regions, and it offers interesting testing ground for looking at the contribution of historical, evolutionary, and biogeographic factors and contemporary ecological conditions into the species community assembly. So in other words, we can test whether historical distributions and biogeographic origins, to what extent they are contributing into the current distribution of the species and how these are being affected by contemporary or less recent ecological environmental conditions. We have several hypotheses about how species are distributed in these zones, but for most insect taxa, we don't really know. For example, we have this model of vertical and horizontal colonization, and we know that at least for some insect groups, the neotropical fauna is in lower and mid elevations, and then in the higher altitudes is being replaced by the fauna of Nearctic origin. So if we look at the habitat, we have a gradient of roughly 2,000 meters, where in the low elevations you can find nice deciduous tropical forest, and as you go uphill, you go through medium deciduous forest, cloud forest dominated by oaks, and then it transforms into mostly pine-dominated temperate forest. So this is how it looks in the lowland elevations. It's a very nice tropical forest with some moderate dry season, and then this is in 2,000 meters altitude, where you have more like typical Nearctic forest. We don't have alpine zone, so the transect or the gradient is relatively short. It's 2,000 meters, but it ends in all the hills are fully forested. And this is the view of our sampling transects. We had 2 of these transects, and on the scale of, let's say, 10 kilometers, there is a change in elevation from 200 to 2,000 meters. So it's like a rather compressed short gradient, which I think is an advantage. And over the several years, we did sampling. We sampled 2 such gradients with 2 replicates per each elevation. This is our standardized protocol of sampling, aimed primarily at ants and certain beetle groups. So this is not the data these are the data which were not barcoded, but they were processed by traditional way. And so we used a combination of Winkler's extractions, bait traps, beating sheets and sweeping nets for understory vegetation, and then directed hand collecting, which is efficient method for collecting ants. And so we get around 2,000 sampling units per each transect, so we have 2 such transects. And the size of the plot is 20 by 20 meters, so it's compatible with what is happening here at BCI. Just some data from this sampling. The ants are behaving as they should. We know very well that the highest richness and species density for ants is in these mid or lower mid elevations between, let's say, 800 to 1,000 meters. But on the other end, their distribution is mostly the best explained by elevation, by also tree species richness, and by tree size. And on the other hand, on the same gradient, we discovered that the abundance and species richness of leaf litter weevils goes in the total opposite with the pattern of ants. So as you go uphill, you get more leaf litter weevils. Yeah, and which are these groups are mostly of Central American origin. They are not from the Arctic, it seems. So in addition to this sampling, we also did malaise traps, not necessarily to sample all the insect communities, but this was like some happy byproduct of this study. And we had 9 malign traps situated between 200 to 1,800 meters with 12 months of exposure. We collected the samples twice a month, and then half of the samples were metabarcoded, and the second half is going to be processed by a traditional way. Few recommendations. It's good not to put the malaise traps close to the bears' pathways, and they really don't like them. And then also during the winter months, they get really curious about this delicious soup of ethanol and insects. So that can affect the sampling sometimes. But otherwise, we didn't have that many such cases, fortunately. The traps were situated nearby or in vicinity of large canopy trees. Yeah, that's one of the aspects which we wanted to cover in our sampling. The main objectives for this study was to look at the richness and composition of arthropod assemblages along the elevation gradient. We wanted to evaluate what is the proportion of the taxa without any genetic reference data. We try to see how different insect orders are affected by the elevational gradient, and whether we can determine which environmental factors are important for the richness and composition of insects along the gradient. And then we also try to look whether we can find any relationship between biogeographic affinity and elevational distribution. And one of the basically primary questions why we started this project was to see how well these malaise traps are good for collecting ants. Yeah, our more experienced colleagues, they claim that if you expose the malaise trap long enough, that it's able to collect maybe 80–90% of the canopy of arboreal ant fauna of the certain spot. So that was one of the things we were trying to test, actually. And so this is the processing pipeline. Pilar Benitez is going to talk about it later today in more detail, I hope. And we processed the large specimens which were of size of honeybee and larger were excluded from the samples, and only parts of their tissues were being used to homogenize the tissue or to standardize for the sample size. Pardon, and then it was grinded. We used the standardized arthropod BF3 and BR2 primers, and was sequenced with CCDB. And then this is the rough analytical pipeline with the sickle, adapter removal spades, V-search, etc. And then we used taxonomic assignations into molecular operation taxonomic unit using the SummaClust, and then compared with or searched through the BOLT and also BOLT and NCBR merger. We assigned this species or OTUs. OTU's assignment was with a 97% threshold, but we also played a little bit with the parameters, but that's the data I'm going to show today. And then 95% and 85% similarity at the genus and family level, respectively. If we look at the accumulation curves and rarefaction curves, it's not great, but not terrible. We got into the about 60% completeness as measured by Chow. We had 50% of OTUs were singletons, yeah, and that's better than some comparable studies from the area. But of course, it's evident that some more sampling should continue in this, to be able to say something more exhaustive about the fauna. If you look at how new OTUs are being accumulated in the sample, so these are the months, and these are the cumulative number of OTUs and a new taxa. So basically, after the 4th month of sampling, we get over 50% of the overall species OTU richness. So which is quite good situation, I think. And then towards the end of the sampling, we did quite low additional or low numbers of additional OTUs. If we look at the identifications, of course, that's something we are going to run a lot during these talks here. We had only about 20% of the OTUs had some voucher reference. That means it was either morphospecies or unidentified species. But if we consider only species identified correctly to the species level with the valid names, it was only 6% of the OTUs, and then 500 were identified to genus level. Yeah, the situation was the best in Lepidoptera because of these long-term sequencing efforts, and then to some extent in Hymenoptera. Yeah, so there is better data available for the region. And the most closely matching data sets with the Mexican one were from Guanacaste, from Costa Rica, then United States, Canada, and some studies from Mexico, which were more limited though. If you look at the composition by insect orders at different elevations, we see quite a curious pattern. And that's somewhat concerning that this low elevation was consistently capturing rather low diversity. We still don't have good explanation for that. The lowland side is the well-conserved forest. And but again, this is the largest limitation of the study. We had only one replicate per elevation, so we cannot really say much about whether there was some bias in the particular site or that something that we would see consistently in other sites of the lowland forest. And the portion of different insect orders was similar, roughly similar along the elevations. If you look at the representation of the most species-rich orders at different habitats, it's always the Coleoptera, Diptera, and Lepidoptera as the most species-rich. And as we go up, the flies, the Diptera are getting more prominent. But the pattern is somewhat consistent at these different type of forests. If we look at by insect order along elevations, then we see that we have several types of responses. Yeah. Coleoptera, Diptera, and Lepidoptera have this hump-shaped curve. In Hymenoptera, we see rather linear increase. And then Pseudopoda and Orthoptera, there are some other types of curves, although we don't have much of the data, we can say. But in, for example, Diptera, it's quite evident that we get surprisingly high richness as we go up in the elevation. Yeah. And the best explanation, environmental variables explaining the richness, were the combination of temperature and precipitation, then precipitation on its own, and also tree richness, diversity of the vegetation. If we look at the beta diversity, it was quite high at both species and genus level. And yeah, we look at beta diversity by different insect orders. It was quite similar for most of them, but Orthoptera and Pseudopoda had the lowest beta diversity along the elevations. So these were more similar between those elevations than the other insects. If we can try to compare some of the environmental predictors for the beta diversity, it was again, for the dissimilarity, it was again combination of temperature and precipitation, and then precipitation on its own. And with the nestedness, it was the good environmental predictor was actually tree species richness. If you look at the beta diversity by forest types, and we just can divide it into these two groups, we see that this temperate fauna is actually embedded in the. It's not very different, but it's embedded into the broader tropical fauna in here. There is a seasonal variation, some rough data. Again, we get some. I would rather like strange pattern. If we look at how many species have been captured in the first months and towards the end of the sampling, then we see that we are at the half of the richness, and we still don't have a good explanation for that. But there was detectable decrease in sampling effort across time. Yeah. And so we can then discuss what it can be. If you look at the actual capture by orders and in different seasons of the year, and then here with the climatic data, we also see there is this decline in what the traps are capturing. And we still need to analyze whether that could be some effect of these extraordinary high temperatures and then high precipitation towards the end of the sampling period. Yeah. So this is that would be. Oh, sorry. Yeah, that would be up there. So. Another thing we try to look at is how biogeographic affinity, or can we say something about biogeographic affinity and how taxa with different biogeographic affinities are distributed along the gradient. It's not really biogeographic origin, because for that we would need like more proper analysis, but based on the known distribution of selected taxa, 200 in this case, we try to assign them into the three principal groups, species with Nearctic distribution, species with Mesoamerican distribution, only located in the South Mexico and Central America, and then species with neotropical distribution, which is anyway from Brazil all the way to Central America. And then what we see is that there seems to be a certain pattern where there are more species with Nearctic and Mesoamerican affinities in the mid and higher elevations, but you can also see these blue lines that some of the portion of species of Nearctic origin or Nearctic affinities are distributed along the whole gradient. Yeah? There is not very good way how to plot that, but that's like a compromise. But if we look at it also by habitat, again, this is based on like very limited unrandom samples of 200 species for which we know at least some data. Yeah, so you can see that actually the portion of the Nearctic species is somewhat similar along the gradient, and what is changing is really the contribution of the Mesoamerican and neotropical fauna, which is decreasing as we go up with the elevation. Yeah. But we see certain portion of species of Nearctic origin distributed along the whole gradient. So it's not really like separated lowlands versus high elevations. We did similar analysis for ants, where we can actually say something about their biogeographic affinity and origin. So this is based on roughly 150 species, and we see something similar. So we see that the species with Nearctic and Mesoamerican affinities are distributed along the most of the gradient, and what is changing is the portion of the species of neotropical origin or affinity. And here we go into the last part of the study. We look specifically at ants and how malaise traps are good for recording them. So these are different methods which we used along the gradient: Winkler extractions, vegetation, understory beating and sweeping, ground foraging, ants based on the bait traps and direct collecting. And then we see that the malaise trap actually recorded slightly over 50 species over the year. And this is just based on the metabarcoding data. And they recorded quite rather unique fauna. So there was not like big overlap among the other methods, yeah? About species coming from other methods. And then there is a similar response by method in terms of species richness. So it has quite similar pattern. So we can see that, or we can say that malaise traps are indeed capturing some exclusive unique fauna, which is not covered by other methods. If we look at the species, ant species by activity, the largest group in these ants captured by the malaise traps were leaf litter ants and then ground foraging ants. There was also a considerable proportion of the arboreal species and species active in the understory. But again, what was somewhat surprising that we get a lot of leaf litter species. Yeah? Again, this can be directly ants crawling from the leaf litter, but it's also likely that these are flying ants during the dispersal stage, yeah, between the which are developing from the leaf litter. And if you look at the composition by trophic level or feeding preferences, most of these ants can be classified as generalists. There was a lot of predators, and then more specialized feeding habits like fungus growings or sugar-oriented diet. Yeah? But again, predatory ants, dorylinae, and generalists were the most present groups in the malaise traps. Just to conclude, some summary. We run into the something which we will hear repeatedly again. There is a major lack of well-referenced reference databases for molecular identifications. We had only 6% of OTU had species level identifications, and about 20% was voucher-based IDs. After 4 months, we were able to get over 50% of the taxa recorded from the whole period. And there has been considerable decline in capture over time. The best environmental predictors of insect species richness along elevation gradient, looking at all insect orders, was a combination of temperature and precipitation and temperature and vegetation richness. We had quite high beta diversity among elevations. And we found out that at least for some of the taxa we could analyze, the biogeographic affinity or origin is not necessarily linked to microclimatic preferences, but contribution of Nearctic species, neotropical species decrease with altitude in some groups. And we also see that Malay traps can capture diverse and exclusive fauna, which is not collected by other methods. Just to continue with the discussion about some more general issues, which again my colleagues have been mentioning, or probably will be in other talks. Something which we should try to address in some collaborative efforts is that there is a limited support for long-term sequencing efforts, which is aimed at building reference databases and collections. I think we also shouldn't really step up our efforts into try to sequence type specimens collections and well-curated museum collections. We shouldn't be afraid of that. And we should try to see how we can facilitate participation of taxonomists in DNA-based studies. And there is still a large imbalance in sequencing efforts between the global north and south, although there have been like some very good efforts and campaigns for these large-scale international collaborations, but there is still quite a lack of the coverage from the most diverse regions. And then, of course, as we talk to our colleagues, there are always these increasing barriers in exchange of materials, which doesn't really help getting the samples around, and due to restrictive or not well-adjusted legislation. And that's something that our representatives or administration should more like be active, right, into inferring these political decisions and drawing the laws which are actually not hindering the science and biodiversity research. Well, with that, I would like to thank you for listening. I would like to thank to our sponsors and colleagues, and thank you for your attention.
Yes, I think we'll go straight into the discussion part of of this first session in the morning. So what we're going to organize, we had a few questions already coming on. It's going to be difficult just to go back to those questions and try to group them by similarity. So what we're going to ask is for the virtual participant, if they have any specific question or general question about, the presentation or just, the problematic of DNA metabarcoding, to simply raise up their hands, and we're going just to give them the right of speaking, and then we're going to see who can answer the question. We should have a micro here for the audience, so we can also in the audience have question. I'm not too sure where is the micro. I might just see. Eduardo, do we have raised hand on? Not yet. Okay. So I will inquire about el micrófono. ¿Tenemos un micrófono? Anybody in the audience has a question, just to start with?
I think Damian has a question.
Okay.
Yes, I do have a question, but I don't know if you can hear me or not.
Yes, we can hear you.
Yes, okay, excellent. It's just a quick question for Daniel. Nowadays, in the metabarcoding and genomic era, doing a phylogenetic study based on traditional barcodes is kind of like being one of those creative taxonomies that you mentioned at the beginning of your presentation, or are they still useful?
So the question was if traditional like single marker or multi-marker phylogenies will become like a thing of the past. I think so. I don't want them
more or less like that.
Like I think we can get now full genomes for non-model taxa, and I think the technology for producing those full genomes is increasing very quickly. I still think that once we have those genomes, we will be able to mine certain markers, and like what used to be a 5-marker phylogeny can now be like 300 or 400 markers comparable across the entire genus. So unfortunately, I think it still depends a lot on money, like how much funding you actually have. The funny thing is that I personally have done some phylogenies with 2 markers, and then I had the opportunity to do it with 100 markers, and the support was identical. Like, okay, not identical. We had better support. That's a lie. But the placement of each taxa on the phylogeny was the same. So I think we do have this tendency to think that more data is going to be better. And unfortunately, the trend is that, like, most people are asking now for more markers, but we're not getting that much more resolution or information. So I think I didn't answer your question, but my hope is that we will still be able to just inform which markers are more informative based on these large whole genome phylogenies, and then you can actually target, with limited budget for those. Does that more or less answer? Yeah.
Seems so. But there is also a price tag. I mean, you cannot so you need to choose. Okay. I have a question for Milan, because it's preoccupying to see, the sampling effort going down. Do you check whether this is consistent among all the groups, or do you know what's happening here? Because that's a bit, worrying.
Yeah, no, I figured that, yeah, you will have some question about that. We really don't know. We discovered it recently in the data. It's consistent among the groups, and it's consistent among elevations, it seems. Of course, if the traps are exposed for a long time, then there is, for example, algae overgrowth on them and so on. But we, for example, in certain intervals, we clean them and put them again. So that shouldn't be the main effect. I also don't think that we captured all the flying insects in the area. And so really without the replicates, we cannot say much. But
because if you think that you have contamination, then you should have more and more an accumulation of beans, not the reverse.
Yeah, no, exactly. So it's there has been some climatic situation. There was extraordinary wet season, a lot of precipitation, so that could have affected the way it can be. It can be artifact on both things. It can be towards the end. There was worse preserved DNA in the soup, so it would be just systematic error in terms of what we are able to sequence, or it would be that really we've been capturing less and less. So we have 50% of the samples are not sequenced, they are there, so we can actually see whether we see it consistently, and that's why I would recommend always have these samples divided, and we can process it by traditional way, or maybe some other better sequencing methods to answer that.
But also in terms of the environmental variable, I mean, if you have, because I guess you're continuing the monitoring, so but if you were to continue monitoring, you could see if, you can recap all those bins, and then if it's really an environmental question. Eduardo, in cabin, do we have more questions from the virtual participant?
Not yet.
Not yet. Okay. Well, any question from the audience here?
Hi, this one is for any of the speakers. I don't know if you have if you have been in touch with other laboratories or such, if they have, or you have been able to find stored, how can I say, samples in order, okay, for instance, whenever you want to see some kind of trends when it comes to climate change, as you all know, you need to compare at least decades, ideally, So the thing is, I'm not such an expert in those things, but I don't know if you have been able to find stored samples in order to compare your results. As somebody said earlier, you can see a declining trend recently, but where the baseline is, I think you already know where my question is going. So I don't know if you have been able to find such kind of samples, and what have you been able to compare or find?
Can you hear me? Yes. Okay. So who wants to answer to this question? Like probably a few people can. Well, of course, we have stored material, but then there is the degradation of the DNA, and that's problematic because then I think maybe Jeremy, you can comment on this. Then you will need other methods to extract the DNA, and that's more expensive. So there is a bit of limitation. Like we have even here on BCI, we had our colleagues Henk Volda, who was actually at the 2 light trap. We were working on in the 70s, from the 70s to the 80s on the island, and then we're trying just to see what we can do with this material. And unfortunately, it's not very well preserved, but we hope still to be able to extract from something, because this is what Daniel said, the baseline year is very important, because for now, I could say probably on BCI, the insect population are relatively stable. I mean, we see some difference. We see that the insects are declining. Some of the are even increasing, because that's probably the immediate response to the slight sort of raise in temperature. That's that's beneficial for most insects, but this is not going to continue like this. It's going to crash down. But the key question is also to know whether other like the baseline that year is very different, and then you might have already had some changes maybe since maybe the 70s, the 60s. This is what has been reported, for example, in Europe, where insect population are probably best studied in the world, like with the Rothamsted survey on other sort of monitoring scheme. And then we see clearly that there is sort of already some huge decline in the 70s, and then there is further decline. So you're right, we should be trying to get all this old material and then try to get more information out of this, but that's difficult. That's going to be dependent on the preservation of the material. I don't know if somebody in the audience, or maybe in the in the whole audience, also in the virtual participant, may maybe comment on the effect of preservation on what you're going to be able to extract as DNA. Jeremy, you want to say something about this?
Sure. I don't have too much to add to that. I would say that most times when I visit a natural history collection for the first time, or an institute like here, part of my tour, it seems, is always to go to the freezers, and I'm shown these samples that are like amazing collections. It's like this great time series. Sometimes they're not in freezers though. Sometimes they're on, shelves in a room that's humid and 30 degrees. And so I feel like my advice is always, let's try it out. Like, find a bit of funding to try some of those samples and see if they work. And then you can really talk about doing more and doing a complete time series. So yeah, I was just visiting Scott Miller at the Smithsonian in DC, and brought a few samples home to Guelph to pass off to the group at the Center for Biodiversity Genomics. And they're Kenyan samples, this wonderful time series that was replicated over a few decades, and you know, we know the material that's been analyzed currently works excellent, but we don't yet know if the samples from 10 years ago and then 20 years ago, I mean, just have to try it out. There's certainly, if they're frozen, that helps in inhibiting the degradation, changing the ethanol. The ethanol becomes acidic over time, so buffering the ethanol or changing the ethanol helps as well. But yeah, at the same time, our methods are improving. The technology, especially around sequencing, is becoming much more sensitive. So I think a lot of those samples can be returned to. We just need to try it out. We just need to more effort in working on these older samples.
Can I add something for that? Sorry.
Go ahead, please.
This is actually came through one of the discussion I had with some of the professors earlier during one of the metabarcoding workshop. So as she asked, like whether can we use like the old samples to interpret maybe like declining of the insects or something, but the question is arising when it's come for the insects. The gene region target is like mostly the CO1. CO1 have like over 500 base pairs, I think around like 600. So when the sample is older and older, there's a higher chance that the 600, like the DNA is getting like a small fragments. So majority of the DNA may be less than 600 base pairs. So the number actually can extract, the species gonna be really low compared with the other taxa who actually sometimes use really small fragments, maybe like 600 base pairs to identify the species. So even if you get some like a DNA extracted maybe for like 30 years ago, 20 years ago, 10 years ago. So even if you see like there's an inclining or like a declining of the species, it's hard to interpret whether actually this is exactly due to climate change or the other factor driving this increase or the decrease, or maybe it's purely because of the quality of the DNA. So it can get mostly, but the question is like, is it possible to interpret the DNA, interpret the results? So I have seen some of the papers. There are some papers actually came for looking at the insect from the old bulk sample, and also those papers also highlighted this issue.
That's quite an observation. Thank you so much.
Yeah, I don't know, if you have experience about sort of decreasing quality of COI sequence.
Yeah, I don't think that's as much of an issue. And I mean, I only say that because we're successful when we work in museum collections, and it's that same issue of degradation. And you're exactly right. The DNA is becoming fragmented over time, so you're just extracting smaller pieces over time, and so that's exactly what you were going to run into in these samples that have been sitting for 10, 20, 30 years, and the poor preservatives that they're in is increasing that fragmentation as well. So definitely methods need to take that into account, and and maybe it's not your standard methods of going after a 416 base pair fragment of CO1, like which was done in the dung beetle paper or dung beetle studies there. But yeah, maybe it's going after mini barcodes or micro barcodes and patching them together. Maybe it involves using multiple markers that may have fragmented less. Especially if you can use a combination that are some from the mitochondria, some from nuclear, some ribosomal, you should see some differences in the fragmentation. So yeah, I think there's some method developments there. I think some validation of those study notes. So certainly those older samples, you would hopefully go through and sort some of them at the same time, and you know, some rough counts of morphospecies or you know, for some of the known samples to make sure that they're on your final OTU or bin list. I think that's necessary, because that, yeah, these types of studies really could suffer from that problem.
But in terms of, again, price tag, you know, Milan has a good point, like what will be good to increase the reference libraries to, you know, to sequence the holotype. Most or many of those, I would say, most common species, they were probably described long time ago. I mean, and then you are usually interested in sort of common species, at least, you know, for the insect ecologists, we are more interested in the common species, because we can do more with them than with the various species. So that's my question is, so maybe for those we're going to have you know, different method of extraction might be more expensive. And my question is, how much more expensive this could be? Do you know a bit? Can you sort of, if it's an order of magnitude sort of higher in terms of price tag, or what?
Yeah, it is much more expensive. Sorry, what was that?
This costs money.
It does. It absolutely costs more money. You know, and it's not always successful too. You know, I think that's why a lot of research groups and a lot of, you know, international programs are working with freshly collected material, because you just don't have those problems with degraded DNA. You're working with high quality DNA, and then you can do, you know, these highly optimized protocols in tiny volumes of reagents and really reduce the cost of it. And that's not possible, or it hasn't been possible yet when working with older degraded museum specimens. And then when you're talking about type specimens, you're mostly talking about decades old, if not centuries old, and so it costs more. If you're going after a 658 base pair fragment of Co1, you might have to stitch it together with, you know, 6 or 7 amplicons. That, you know, there's a lot of issues with just amplifying, you know, fragmented and degraded DNA. So maybe amplicon sequencing will not work for particular specimens, and then it's shotgun sequencing is and genome skimming is a better strategy, but then that's of course more expensive than you know, sort of standard amplicon sequencing. So I think now, you know, we've been talking this week about Rodolphe Meyer's lab and the amazing work that's being done there to use nanopore sequencing and to really drive down the prices in that way. And certainly the Center for Biodiversity Genomics at Guelph has always made that a priority to get prices as low as possible and kind of extend that to the scientific community. So I think, you know, if you're running a specimen that you collect today or over the last couple of years, you know, you're kind of talking in the $1 to $5 range. You know, sometimes lower, sometimes a bit more. And then And then if you're doing museum specimens, which are kind of 2 to say 30 or 40 or 50 years old, then it's probably in the, you know, 10 to 20, $25 range. And then if you're talking about genome skimming, I think that's, you know, I think the current rates, at least in the US, are probably sort of in the $50 to $70 to $100 range to do a sample. So you can see that step up every time. And what I might mention in my talk this afternoon is that, yeah, I mean, specimens fail, so you might want to have some redundancy in your sampling. If you're working on types, then maybe you want to do, you know, several of the paratypes just so that you get one of them to work. So you have to consider that in your costs as well.
Just to finish on the types, so do you have practical experience? Like, what was the, you know, the response from the taxonomic, you know, community? Are they sort of quite responsive positively, just in basically cutting a few legs of maybe not the other type, but the paratypes and so on? What's sort of the general response? Is that difficult?
Yeah, it can certainly be challenging. I have been fortunate to work with Scott Miller again, who might be online, with Axel Hausmann at the ZSM in Munich, and they have been very proactive in this on this subject, and know the importance of using types when possible. So they have been able to, within their collections, to liberate these type specimens for these types of projects. And then I've also been really lucky to work with fantastic people at the Center for Biodiversity Genomics, like Sean Prosser, who was and is just a whiz when it comes to somehow finding the DNA, you know, in these 200-year-old specimens. So my experience in it is positive, but I've certainly heard of other stories in other collections, where researchers come in and try and work on types, and it's not successful, and then you know, there almost always has to be some sort of damage done to the type specimen. So if there are no results and there's some damage, then maybe the reputation of type specimen barcoding is tarnished a little bit in that collection. So that certainly happens too, I'm sure. But I do feel like I feel like there is a lot more priority placed on freeing up collections for barcoding and other genomics work, and curators themselves are certainly, you know, heavily involved in this, and they want to do it too. So I think there's a lot of collections that are opening up their type collection for this, and putting in some protections too, because it certainly shouldn't, they shouldn't be opened up to be to be harvested in a way that might damage them permanently.
I think we have some question from the virtual participant. Okay. Well, who was first?
Luciano was
Hi.
first.
Yeah, thank you. Thank you very much for all the presenters. Very amazing presentations. I was having the same doubt about how to standardize different samples among seasons. So I would like to know, based on your experience, which type of metadata would be the most useful to correct for false negatives between samples. I don't know if maybe there are some conditions in the field that will in a particular season that will affect more the degradation of eDNA, for example, or DNA, overall DNA in a bulk sample. Yeah, both in, I don't know, in field and in the laboratory. Yeah, by analyzing the purity maybe after DNA extraction. I would like to know which variables would be the most useful, or do you believe are the most useful? It's an open question.
I think I can tell you what we did for the light trap and the Senacid. We had like a bunch of different methods, and all of them have some issues with the catch. So I think one of the most important things is that you are on top of your ethanol in your trap. So for pitfalls, those that have, like, let's say, open containers where you can get a bunch of debris and it can evaporate or it can be diluted by water, you should really be on top. So I remember pitfall trap, we had to go every third day to change the ethanol because that one was super dirty. Then for the Berles and Winklers, there is extra laboratory procedures to clean the soil. So that's an extra step in the lab. So if your catch is always with pure ethanol, you would be okay. You can also protect your traps from the rain. And more or less, that's what you can do in the field. But then in the lab, what the Canadian Center did for us is to actually do aliquots of your collection into 3 replicates and sequence each of those 3 replicates per trap with positive and negative controls. So I think, it's a little bit out of our hands how the DNA degrades, but we can ensure that the liquid or the ethanol where it's actually being collected stays as clean as you possibly can. And then replication, just try to sledgehammer your data so you can actually, you can then have certain controls, positive and negative, and you can start looking if this one OTU or bin occurs in the 3 of your replicates, you can be pretty sure that it's there. If it occurs in 1 of your replicates and in your negative controls, well, probably a false positive. But there's a bunch of techniques for correcting with that. There is some even R packages that use your negative and positive controls to measure a proportion of reads, which could be, so if they're present in your controls, they could be a contamination, but it takes a proportion of reads to actually statistically decide if it really is contamination, and then you remove it from your actual samples. Other people have like a bit more of a blanket approach where if it's below a number of reads, some people have used 10 reads, some people use 100 reads. If it's below that, you just remove it. That's super conservative, and I think it really helps you to take care of false positives, but you can suffer by taking away a lot of the false negatives. So it depends on, again, your question and what you want. For monitoring, because you're more interested in common species, it's okay if you lose a few false negatives, but I guess you don't want to. But yeah, I think it's also developing. I don't know if there's more lab work that you can do to ensure. But in the field, the only thing that we did, and I think the thing that works the best, is to control your ethanol. Like, you don't want it to evaporate, you don't want it to be diluted, you need to change it from time to time. And I think that's what I would say. I don't know if anyone has other opinions.
Maybe you can give the, we have a question from Scott Miller in Washington.
Oh, hi, this is Scott Miller from the Smithsonian in Washington. And I just wanted to follow up on Jeremy's comments about access to collections, because I am both someone who extensively mines collections for DNA, and also I'm a collections curator and responsible for their care. And I think the key thing to keep in mind from the standpoint of the museum is that the museum is charged with both the preservation forever of these specimens, and it's also charged with making them accessible for use. And that involves, weighing the costs and benefits. So I think when and there are some museums that are very liberal with access to DNA sampling, and there are others who are unwilling to even consider it, and a lot of them in between. And it gets particularly sensitive when you get to holotype specimens, because there's only one of them and they have historic value and all of those other things. So if you can, in some cases, there's paratypes from the same locality or collecting event as the holotype, and that's sort of almost as good. So there's ways that you can work around that sometime, but I think when you approach a museum collection, you need to be able to demonstrate the success of your method. And we are having amazing success through Guelph with pinned insects that are 100, 150 years old. Not every specimen works, but an awful lot of them do. The museums may be concerned about the handling cost, because somebody's got to find the specimen, get it out of the collection, take off the leg, put it back, do the imaging, all of those things. And then I know a number of museums have had trouble in actually getting the data back from people who have sampled DNA from their specimens. We have some of those problems. So, being a good collaborator and sort of setting up the conditions of the exchange of services, if you will, is important. And I would also say, finally, there's kind of two ways to do library building. I mean, a lot of what we do at the Smithsonian is, the things that Jeremy is going to talk about this afternoon, where we, have shopping lists and we're trying to get every genus or every species to the extent that we can. But for ecological projects, one can also do more targeted library building if you know that, certain lineages of insects are eating your plants. One can go in and try to build a library of things of that genus from whatever country. So I think it can be done in a cost-effective way, but it also, I agree with Milan's comments. It really needs to be geared up and done at a global level. That we know how to do it. We just need the will and the funding to do it. So I look forward to Jeremy's talk this afternoon.
Well, thanks, Scott. I would say that, for the mining the museum collection is going to be expensive. And as you say, you can target different genera, so on. But for ecological studies, again, you're probably mostly interested in common species. So we can actually do it cheaply by simply, sequencing fresh material. So it's a different sort of, I would say, strategy. I don't know if we have other, do we have other questions from the virtual participant? Do we have other questions here? I must emphasize, maybe in the audience, in the auditorium, a few people speak Spanish. So if you want to ask questions in Spanish, please do so. And then we'll translate the question and translate the answer. Any question, anything? If not, it's already past the time, so we are going to have an hour for lunch, and so we'll come back at 1 o'clock. And thanks to all the speakers for the session, and then for everybody who's been attending this meeting. Thanks.